will be hearing 3 presentations, and 2 of them will be from 2 sisters, but they're not literally sisters, but they're sisters in brotherhood and sisterhood. So we will be thinking, we're going to hear 3 lectures, about 20 minutes long each, and they will all be thoughts prepared by students of Theosophy from different traditions. And the subject will be, what is brotherhood, what is nucleus, and what is healing? Kim is a longtime student at the United Lodge of I.S.I.S, and she will share her thoughts.
Well, thank you. I think I'm going to just tilt it here. I thought I'd talk about what is a nucleus and leave everything else for others. So, talk about the little I know. When we think about what is a nucleus, I think in common speech, we have been sometimes saying it's like a seed, perhaps like a nucleation site of a crystal forming. It's the beginning, the source. And that's a fruitful way to consider the concept of a nucleus. We could think of a crystal forming in a supersaturated solution. In order for it to form, it requires a certain organized setup of atoms that form an ionic compound that then propagates and grows larger, forming a larger and larger crystal. This is following a given pattern created by the organized structure of the initial ion complex. That's one way to think of a nucleus, as like a seed in a crystal. Another, when we speak of a seed is not a nucleus. And not that we need to get too literal on everything, but there is the idea of as above, so below. And I thought it might be really interesting for us to think about analogy and correspondence and really investigate the difference between a seed and a nucleus, and then go further deep into what is a nucleus. So if we think of a seed, for example, a bean seed, it has basically 2 parts. First, an embryo, a tiny adult form, a tiny baby plant. Second, it has a lot of food supply to nourish that embryo. Some seeds are very small, like a carrot seed. Doesn't have much in the way of supportive nutrients, but it has that embryo inside of it. Whereas larger seeds are generally larger because they have a lot more nutritive substance in them. Again, a seed then is made to encapsulate an embryo over a long term through harsh conditions and preserve that future generation, possibly for thousands of years. Some seeds have been known to be viable after several thousand years, which is quite astonishing if you think about it, that there's an embryo of that plant, of that living being, held in stasis, almost like a spore. But H.P. Blavatsky did not say a seed of brotherhood. She said a nucleus of brotherhood. So I'll take her at her word. Nucleus, not a seed. So a nucleus is different than a seed. It's an organizational location of active control of a cell. It's active. It's not in stasis. It's not holding something for the future. It is actively controlling all functions of the cell. And it's really quite fascinating. So I am going to quickly show you, here's a nice cell biology diagram from the textbook. And that large purple thing on the right is the nucleus, right here. And I know that we could get involved with lots of cell biology from high school, and due to time, I'm not going to go into all the details, but what I think is important to know is that all cells of organisms that have more than one cell have a nucleus. There are life forms that are one cell Many of them do not have a nucleus. And they're very simple little bacteria. All of their cell functions occur inside their cell membrane of the outer envelope of the cell membrane. And this actually creates a kind of confusing environment because a cell, in order to function, needs to do things. It has to do things using proteins. It has to make proteins in order to function. To make proteins, the chemical environment for making proteins is very different than the chemical environment needed to create the energy to run this system. In cells that are more advanced than those single-celled organisms, what are called eukaryotic cells, they've segmented the operations of the cell so that the energy-making portion, which is highly oxidative environment, very reactive, is encapsulated into the mitochondria, a subsection right here. And the protein manufacturing section of the cell is encapsulated in here. It's called the endoplasmic reticulum. And if it's a plant cell, it has chloroplasts, which are also little organelles that are designed to take energy from the sun and turn it into highly energetic compounds that are useful for the rest of the cell. This structure of the cell enables there to be protective barriers within the cell so that chemicals that are very strongly oxidative and might react with other chemicals won't do it. So they'll be in their own space. This is where the nucleus comes in. The nucleus is necessary in these advanced cells, which are in dogs and birds and worms and humans and elephants. These more advanced cells, the eukaryotic cells, have a nucleus right here to protect that very important informational molecule called the DNA. And here's a larger nucleus for us to look at. It has a membrane around the outside. This is dark purple. That membrane separates it from the cytoplasm of the cell, that goopy material that is all throughout the cell, and the inner part of the nucleus. The inner part of the nucleus has actually a lot of structure to it. It's not just sloshing around in there. It's got some little fibrils called chromatin and a structure that enables the DNA to be very highly controlled and unwound in order to be replicating, and then also transcribed, copied for the other molecules that transfer that information to the outside of the nucleus. There are pores, these holes in the nuclear membrane, and those very tightly control what comes in and out of that nucleus. It was mentioned earlier that there's a nucleolus inside of some nuclei. That's right here. This is where there are a lot of ribosomes being manufactured. Ribosomes are the protein structures the RNA structures that create proteins. And then we have outside here the ribosomes that attach to the endoplasmic reticulum. I don't want to use a lot of words here, but basically, proteins, that all-important stuff, is made here. And the nucleus, being a protective location for DNA, is something where I think it's helpful to actually look at an animation, and this is where I'm going to get some help right now. Go ahead, Erwin. We're going to look at an animation of how DNA actually functions in the cell, because I think it's highly suggestive. Through analogy and correspondence, I think that we could have fruitful intuitions about this whole process. So, why don't you just go ahead and start running the 1 through 12 here. So here's a cell. This is a plant cell. I'll keep going. And inside the nucleus, we have chromosomes. These are tightly bundled bunches of DNA. You can go ahead. And those DNA molecules can be unwound and looked at more closely, and you can see the double helix pattern to them, which is highly suggestive. Why this double helix? Very interesting. Go ahead. And this strand of DNA cannot get outside the nucleus. The nucleus is where it's kept, and its information has to be transferred out of the nucleus to make a protein. How does it get that information out? Through transfer RNA. Go ahead. So the code gets read. Go ahead and do the next one. There are enzymes that go ahead and unzip the DNA and read off the pattern. And as they read off the pattern, they create a little messenger molecule. Go ahead. That's called transcription. That little messenger molecule is small enough to go outside the pores, and it goes into the cytoplasm where it enters the place where a protein will be made. Go ahead. And go ahead. The purple things there are building a protein molecule off of the message that they just received from the nucleus. Go ahead. And then that protein folds up and becomes a usable item, which then controls other functions of the cell. So maybe if this is a hair cell, it's going to be making hair. If it's a muscle cell, it will be making more of that material that can allow the muscle cells to work. If it's a nerve cell, it would be creating neurotransmitters. Go ahead. And then it travels to where it needs to go. Go ahead and do the transcription now. So that was just an overview in the middle of that blue. Yeah, thanks. So here we're going into a little bit more detail. Go ahead and hit next. So actually, we can go pretty quickly on this part because I don't want to get too much into this. So there's different regions of the DNA that are read by the messenger RNA. And cell biologists are finding out a lot of fascinating things right now. This is an incredibly fast field of research right now. They say the half-life of knowledge is 2 years, meaning that if you graduate with a degree in biology, in microbiology, then in 2 years, half of your knowledge will be irrelevant because the advances are so quick. In the Human Genome Project, they analyzed the entire genome, and through studies, they've found that there are specific regions that enable the control of the DNA's replication to happen, or the transcription. This is the fascinating thing is, you can have DNA with all sorts of information, but what is being used right now? That's being controlled by molecules who were themselves made by the DNA. So the products of the DNA regulate itself. It's very interesting. Yeah, and just go ahead there. So here's the enzyme that reads the RNA. You can go ahead. I think actually this is, keep going. Yes. So keep going here. I think I'm trying to just do this quickly here. So it has to unwind in order to be read. And in this section, the DNA has a certain pattern of nucleotides. The red RNA here is reading the opposite. It's creating a mirror image of this strand of DNA. So these two strands of DNA are, in a sense, mirror images of each other. Since the bottom DNA strand is being read, its mirror image, this red mRNA, is the same, basically, as this other strand of DNA, because they're both mirror images. This is a mirror image of each other, and these are mirror images of each other. Go ahead. And here we see specific letters, A, T, C, G, A, and sometimes U here. Those are symbols for different shaped molecules called nucleotides. And we have adenine, thymine, guanine, and cytosine, just symbolized A, T, G, C. And the interesting thing is all life has these same nucleotides. Viruses and also RNA has U instead of T, but that's a trivial point. The main thing is, all life has these nucleotides. I think that's quite fascinating. The DNA is made the same in a cat and in a crocodile. They're the same. Okay, go ahead. So here's, again, the little strip of polymerase. Go ahead. We should go to translation. Sorry. Quickly, we'll just do that. Yeah. Okay. So here, this is the next step. After we've made the messenger molecule, go ahead. Messenger RNA, that little strip of red, is going to go out of the cell, out of the nuclear envelope. Go ahead. Through a pore, and then it goes to the manufacturing place. Go ahead. Where, keep going. There's the ribosome. It's going to go through there. Go ahead. Keep going. There's a whole bunch of loose amino acids. These are the building blocks of proteins. This is the powder stuff that people take to be bodybuilders. Amino acids. Go ahead. Yeah, and so here's the information sequence. It's pure information. It's useless, in a sense, in that it doesn't do anything except have information. It is a pattern. This is the ribonucleic acid. Nucleic acids only have information as patterns. Patterns to be mirrored in the protein world. Go ahead. And there are groups of 3 nucleic acids that form a codon, and these, keep going, will be read by the the transfer RNA. Go ahead. And they have specific patterns to start and end. Keep going. The nice thing is that floating around in this cell is amino acids, which will build up the protein, connected to a little pattern which will fit exactly on that messenger strand. Keep going. So that little codon is going to fit onto the strand. The anticodon fits to the codon. Keep going. There, it's going to fit right there. Keep going. And moves on to the next. Keep going. And then here, this CGU is going to be matched up with the next anticodon. Keep going. And eventually, you see down here, these amino acids get stuck together. So the pattern gets translated into a pattern of amino acids. The nucleic acid reflects and becomes amino acid pattern. Keep going. And there they are, reacting and bonding together at the bottom. Yeah, so this is called protein synthesis. And every cell does this. Yeah, keep going. Yeah, keep going. So we end up with hundreds of chains of amino acids strung together to form proteins. And there they are, just going longer and longer. Now, this is really nice as an animation, but I have a short video to share of the discovery of this process. I thought I would share it with you. There's 2 short videos, and then I'll just say a few things about them. Yeah, so start at the beginning, I guess. Where they were going to share their insights. Yeah. Much of their work was based off of Rosalind Franklin's work as well. She didn't get as much credit, though. But yeah, I included that because of the awesome, the wonderful animation of that DNA and showing it unwinding and unfurling with its replication. It's astounding. I think due to time, I'm not going to show the second animation. Am I right that I should be quitting pretty soon? Yeah? Okay, just a few words to end. DNA is one of those symbolic elements that reminds us of the first fundamental, the absolute truth, in a sense, or the absoluteness, be-ness, perfection, transcendent. It's transcendent because it's unavailable to the rest of the cell. It's beyond the cell. It's in the nucleus, abstract, untouched by the machinery of daily life. It's, in a sense, like the Pure Land teachings of the Mahatma. When those abstract teachings come into the world, they have a vehicle, the vehicle for translation. That vehicle in cell biology is mRNA. It goes through the membrane of the nucleus into the realm of activity of the cell. It's like buddhi. That messenger is like the divine ray coming from that absolute that then is embodied in such a way that can be useful. To be true and useful, it has to be true to that original pattern. That's instructive for us. Buddhic intuition is always mirroring the divine truly, inspired and undefiled by what it enters into. That Buddhic intuition, like the mRNA, goes into that cell environment and interacts with the higher mind, like the tRNA, that can accept the message of that Buddhic ray in such a way as to translate it into the materials of the day, the materials that are there, the amino acids. Those materials that are there become useful structures for the cell's functioning, for the cell's active functioning. A cell is responsive to its environment. It is not statically exuding and without any response from the environment. It is dynamic, and the nucleus is responsive to that need of the day, of the moment. Hormones or other cell signals, the environment, may cause the cell to change what it's creating in the nucleus. The nucleus may transcribe different elements based on the needs of that particular moment. So, in a sense, we could see the three fundamentals acting here. We can see the first fundamental in the cell DNA as being needed to be absolutely perfect and to have total integrity in its message. The second fundamental we see as periodic flow, cyclic law. The nucleus is constantly involved in cyclic patterns of of creation and destruction, of flow out and flow in the sense of the DNA is constantly involved with either replicating itself or pulling together tightly in the form of chromosomes and undergoing division. When it divides, the cell nuclear envelope dissolves. So the nuclear envelope dissolves, the cell splits in two, then the cell nucleus reforms again, and the DNA unfolds again and begins to replicate again. So there's always this flow of replication and then congealing together for the cell division. And then the third fundamental, that of being, that there's this divine pattern going through into the manifestation in the environment of the cell and dynamically creating all of the molecules needed for life. Incredible diversity of structures can be created. All the various living things on Earth are created through all their diversity, through this process of translation of the DNA's message into amino acids and proteins. So in a sense, that's like being, the third fundamental. And I think that Voice of the Silence really says it right when it says, compassion is no attribute, it is the law of laws. If we ask what is that inherent nature of the DNA, it's common to all of us. Common to every living being on planet Earth. That pattern of A, T, G, and C. It's like compassion, the law absolute. So, there's more I could say, but there's many more to hear. Thank you.